All right, let's go ahead and get started. Good morning. My name is Mary Leonard. I'm chair of the Department of Pediatrics, and it's my great pleasure to welcome you to this morning's Pediatric Grand Rounds. As you can see, as always, we have a wonderful lineup. Wishing everybody a very happy and safe Thanksgiving next week. And then in December, Dr. Chou will speak with us about hypnosis in the medical setting. And we're just thrilled to host Dr. Madhu Misra from Harvard to speak on oligoamenorrhea in athletes. She's really a thought leader in bone and body composition and all sorts of metabolic disorders and eating disorder and athletics. So it's going to be a very exciting Grand Rounds, and we're delighted to have her. Please make sure you have the correct text code here to get your CME credit. It is different every week, and we'll make sure for those of you who are just getting settled in that it's in the chat box as well. Next slide, please. And then we also want to draw your attention to, I hope everyone's very familiar with the COVID-19 in Children seminar series. It's really an extraordinary contribution from our faculty that have put this together and absolutely keeping the most up-to-date and important information in front of us. There's been a great deal of information in the news about the impact of COVID on the heart. And this will be really exciting to hear it from the perspective of our pediatric colleagues shown here. Next slide. Okay, so I'm going to pass it off to David Moss, who's going to introduce Dr. Chou. I just want to say, David, I'm sure is well known to everyone here, is our chief of pediatric endocrinology, and his list of accomplishments in the 4 years since he joined the faculty, I think we just had an anniversary, David, is really quite extraordinary. But one of the most exciting things that David has done is bring just incredible talent into this department. Recruiting Dr. Anna Gloin and now with Dr. Chou. One of the things that I love about the work that Danny does is really a perfect example of the strengths at Stanford, where our faculty collaborate not only with people across the School of Medicine, but across the entire campus. And when we were first bringing Danny here, we realized that this was an opportunity to forge some really fruitful partnerships and collaborations with faculty in chemistry, biology, material science, and engineering, and bioengineering. So thank you, David, for bringing Dr. Chou here, and I'll leave it to you to make the introduction.
Thank you, Mary. Thank you for the kind words. It's greatly appreciated. So it's my pleasure to introduce Dr. Danny Chou, who joined us this spring. Danny was initially trained as a synthetic chemist, working on small molecule synthesis during his PhD at Harvard. Then, as a postdoctoral fellow at MIT, he worked with Daniel Anderson and Robert Langer, and he developed glucose-responsive insulin derivatives for blood glucose control. He then began his independent lab career at the University of Utah and continues work now at Stanford with a focus on developing peptide and protein therapeutics for human diseases. Danny's been funded by the Department of Defense, the American Diabetes Association, and currently by the JDRF, as well as multiple NIH awards. So we're quite pleased to have Danny as a member of the division, the department, and the Stanford community. Also, as a spoiler alert, he will be showing us a video of a cone snail eating a fish. So you're going to have to stay tuned to see that. So Danny, we're excited about your talk and really excited that you're here on the team. Looking forward to the talk. Thank you.
All right. Thank you so much, Mary and David, for the kind introduction. My name is Danny Chou, and I'm pretty new in the department, and we just joined, well, theoretically joined in April, but because of pandemic, so our whole lab moved here by the end of August, and then we are pretty excited to join this community. Today, I'm going to talk to you about some of the efforts my lab has been doing in the past few years to basically using insulin engineering to develop new kind of insulins that can potentially achieve optimal blood glucose control. To begin with, I'm going to show you basically the traditional insulin development timeline. So I think next year you will be 100-year discovery of insulin originally from University of Toronto. It's been a long way since the initial discovery, and then there are lots of achievements throughout the past 100 years. So basically, in the old days, people are still using various insulins from animal origin to treat people with diabetes. But I would say starting in the 80s, because of the DNA recombinant technology, we started to have biosynthetic human insulin. And then because of that, people can start to do some mutations in the original insulin molecules to achieve different pharmacological properties. So more on the basically how to make insulin more rapid acting, which I will talk about later, and also how to make insulin to stay in your body for even longer time. And then basically throughout the time, people are just trying to make things to be better and better. So this is a review in 2015. So at that time, the inhaled insulin Afrezza just got approved. They are predicting a bunch of different rapid acting ones. And then this one, in the end, called Fias from Novo Nordisk, they got approved. And then I think some of the ones here, they will be approved soon as well. But these are basically still using the same fast-acting insulins are developed in the 90s, but they are changing some of the excipients in a way to make them to be absorbed faster. On the other hand, in the long-acting insulin part, still there are progress to make things from daily use to more than daily use and potentially to weekly use. But one of the exciting area is to develop this thing called smart insulin, which is a catchy term of a kind of more general way for us to say that, which is glucose responsive insulin. So today I'm going to talk to you about two of our efforts that we are trying to do to basically develop better and not high the smart insulin, and how to develop even faster acting insulin. So this is just a very kind of general way kind of to show you how normally when people with diabetes, they will use insulin to modulate their blood sugar level. So basically for healthy individuals, every time when we are eating something like 30 minutes ago, I just had my breakfast. And because I'm eating something, my pancreas will just secrete insulin. So basically, I don't really need to worry about what am I eating in each time of the day, because my pancreas will just adjust to it and then basically secrete the right insulin amount to modulate my blood sugar levels within that perfect range. And then for people with diabetes, unfortunately, this effect is gone, especially for people with type 1 diabetes. So normally in the old days, before the approval of insulin pumps, people normally need to inject a basal level of insulin in the beginning of the day, basically to provide the basal level need for the day. And then throughout each meal, you will pretty much calculate what's the amount of calorie and then different type of food you are using, and then to decide what is the precise amount of insulin you will inject there. And then the concept of glucose responsive insulin, people have been thinking about it, I'll say since the 70s. People are thinking about whether we can develop something in a way that this thing is glucose responsive. That means this insulin, using whatever approaches or designs you are trying to do it, but the activity or availability of insulin will be basically in response to the circulation glucose levels. So if you can do something like this, then that's great, because that means it will just be like the beta cells in the pancreas in the way that you will only secrete insulin when you need it. So in this case, you can imagine that the blood glucose levels can be tightly controlled. And then potentially this is something that you can greatly improve the convenience in a way that you don't need to do multiple injections per day. So try to do this to develop glucose responsive insulin. Normally you will need some kind of glucose sensing element such that the whole thing can sense the glucose. So one of the common one to use is glucose oxidase. Basically, this is an enzyme that will convert glucose to gluconic acid. And normally, you can either use this acid to generate some kind of mechanism. So as you can see here, this when you have more glucose, you will generate this acid such that this kind of polymeric system will be dissolved or swell, and then that will lead to insulin release such that more insulin will be released when there are more glucose. And then another mechanism people can use from that is because in this process, this hydrogen peroxide will be generated. So this hydrogen peroxide can be used as some kind of oxidation signal or some kind of electrochemical signal during the triggering. So some of you may use glucose meter quite a bit, and these glucometers, it basically use this hydrogen peroxide mechanism to know what's the blood sugar level there. And another approach is to use a small molecule called phenylboronic acid. And basically, this is not an enzyme compared to glucose oxidase. And then it's actually pretty straightforward in a way that basically this is a molecule such that when you have more glucose, you will shift the chemical equilibrium from an uncharged state to a charged state. And then similar as the previous system, if you can put your insulin inside some kind of hydrogel or polymeric system, when you have more charges because of the glucose, then these charges, they will repel each other such that the polymer will swell and then insulin will be released out. So basically, in these two examples, I'm showing you I would say, very typical way for people to achieve glucose response to insulin, which is to put insulin inside some kind of gel or coverage. This is pretty straightforward to do because it's pretty easy for us to contain insulin in some kind of coverage setting, and then basically you will use some of these mechanisms to achieve that. But there are pros and cons. It's easier to achieve, but the con side is that you will also need those kind of polymeric coverage to be injected together. And then we know that diabetes is a chronic disease. So that means if you are doing the injection every single day, you will also need to inject the polymeric system every single day. And then that also basically coupled with some of the potential immunogenic effects. So I would say that this kind of polymeric system is not really well pursued in the clinical development. So how can we achieve all these things without using the polymeric system? That means we will need to basically incorporate these glucose sensing mechanism in the insulin by itself. And this is something I started to do when I was still a postdoc at MIT, and then this is a mechanism we go for. So one of the long-acting insulin called insulin datamere, its long-acting mechanism is it has a fatty acid side chain, and this is a pretty hydrophobic side chain. So you can imagine that when you have a side chain like this, it will interact with the hydrophobic pocket of C1 albumin, which is we know the most abundant protein in the blood serum. So basically, what we are trying to do is, if we can incorporate this phenylboronic acid that I just introduced to you in the previous slide into this fatty acid chain. So you can imagine that now, when I have more and more glucose, I will make this insulin datamere, or the modified one, to be more and more negative charge. A negative charge will make the whole thing more hydrophilic, such that this insulin, when there are more glucose, you will release from albumin. Okay, and then this work is published already. So I'm just going to go quickly to kind of show you what this kind of thing can achieve. So this is what we use. Continuous glucose monitor. I mean, these are basically the same kind that people with type 1 diabetes, they are using to monitor their blood sugar level. So basically, we just connect them into the backside of the mice. So you can see that we can basically get the sugar levels every 5 minutes. And then so these are, so the first graph here, these are a group of healthy mice. So so you can see their blood sugar levels are in the perfect range. Around 3 hour-ish, we inject glucose in the IP space. So basically, it's just to mimic the meal intake process. So you can see that because the pancreas is healthy, and then so you can see that basic glucose level will go up and but very quickly go down. So this just the perfect control one can imagine when you have a healthy pancreas. This second group, the blue line here, these are diabetic mice. So basically we do that by injecting a drug called STZ. This is something that will specifically kill beta cells in the mice. So you can see that their initial glucose levels is in the hyperglycemic range. And then now we inject the long-acting insulin. So basically this is the control of insulin determiner. So basically this is long-acting insulin. So when you inject it in time 0, so you can see that glucose level will slowly go down to the normal level, but and then also around 3 hours, you inject glucose in, glucose level will go up, but you still kind of go back down very slowly. And this is basically the typical situation if you do not have a specific fast-acting insulin around the mealtime to address it. Basically, you will spend more time throughout the day in the hyperglycemic range, and things like this ultimately in the long run may lead to certain kind of complications that's going to impact the health for these people with type 1 diabetes. In the case of these PBA modified determiners, so you can see that still in the beginning it's in the hyperglycemic range, but when you inject insulin in time 0, the slope of the blood sugar level to go down is much faster. It's much faster. And then basically around 3 hour time, we also inject the glucose in, so you can see that glucose level go up, but the glucose level will still go back down pretty fast. And basically this is to show you that this is because these are potential glucose responses in insulin such that when we have higher glucose, like the previous slide I show you, more insulin will be released from the albumin depot such that we can achieve the optimal glucose control by such design. We further use the glucose clamp studies to demonstrate the in vivo activity of insulin. So for some of you who are not very familiar with glucose clamp study, I mean, these are pretty typical thing one can do on humans, but with the right surgery technique, we can also do that in mice. So basically, what we are trying to achieve here is basically we want to basically make the glucose level to be somewhere around 350 milligram per deciliter, and then so basically you just put the insulin in such that the insulin will be active, and then what you are trying to do is to basically to see whether you can infuse in the glucose to the amount to always maintain the glucose level inside the mice to be 250 milligram per deciliter. So the more insulin you need to infuse in, as you can see from the panel B, the glucose infusion rate there, the more you need to infuse in, that means the more active the insulin is. So basically, so you can see that in the normal glucose range, there's not much difference between determiner and the glucose response to insulin. But when you clamp them in around 330 milligram per deciliter, so you can see that there's a 40% excess of activity for the modified one compared to the controlled long-acting insulin one. So basically, we can demonstrate that basically in such design, we can see a 40% increase of activity to go for that. And then when I started my own lab, I was thinking about another potential design that could be useful to further make the whole glucose response insulin performance to be even better. So this is do another long-acting insulin called insulin glargine or Lantus. So Lantus is another long-acting that I would say endocrinologists prescribe a lot, and then it uses very different mechanism to achieve the long-lasting property. So it actually, compared to the determiner, it doesn't have a fatty acid chain. All it does, I mean, the major difference is it has 2 more arginine in the end of the insulin B chain. So what does this do is these 2 arginine will change the so-called isoelectric point of this new insulin protein to be somewhere close to 7. And what it does, you will make this insulin, Lantus, to have very low solubility around physiological pH. So what happened? So what happened in the end? Let's see. Okay, so what happened in the end is when you inject glargine into the body subcutaneously, you will basically have them just crash out, because it normally will dissolve quite well in pH 4 because its isoelectric point is closer to 7. So it's very concentrated, but injected in, now its solubility in physiological pH is very low. So you will crash out as more of a crystal type of molecules there. So now you will slowly and sustain release from the depot, and then such that it can basically achieve the kind of long-lasting property. So what we did here originally is actually very similar to the previous concept, but basically when you put the phenylboronic acid there, in the end, you will not change the isoelectric point. But when now there are more and more glucose binding to the boronic acid, now you will change the isoelectric point because there are more charges to increase the solubility. So now, as you can see from panel C, now we can potentially expect that more and more smart glargine will be released when the glucose level is higher to achieve the glucose stimulated release compared to just a normal slow sustained release here. So this is to show you that all these modified insulins, they pretty much have the same same bioactivity. There's no much difference. And then this just to confirm that yes, we are indeed seeing a different glucose-dependent solubility. For the control insulin glargine or Lantus, its solubility remain the same when glucose level is low, normal, or high. But in the case of the smart glargine, so you can pretty much see a linear type of increase of solubility when the glucose level increases. This is the same glucose clamp studies I mentioned to you previously, but now we are doing something even more. So basically, we are clamping them in the normal glycemic case and clamping them in the hyperglycemic case to see the difference between the two insulins. And here's the result. When you clamp them in hyperglycemic case, so this is somewhere around 400 milligram per deciliter. This is the very high glucose levels you want your insulin to be active. And then so you can see that the activity between the two insulin, they are very similar. So that's great. That means that both insulins will be quite active. In the 100 milligram per deciliter, this is already the normal or the euglycemic range. You don't want your insulin to be too active. So you can see that the smart glargine is only 30% of the activity compared to the control insulin. So this is great. And then this means that if this is the case, that means, you know, normally one of the constraints of insulin injection is overdosing. So if you don't inject enough, that's okay. You can, you know, inject a little more later. But if you inject too much, that will normally lead to very severe consequences, you know, potentially in severe hypoglycemia. So if you look at this data, if this is right, that means in 100 milligram per deciliter, this, you know, smart insulin is very low activity. So potentially I can inject quite a bit without the worry or danger of overdosing. So that's what we try to test here. And then here's the result. So basically, we can basically inject the insulin in, and then we are not seeing much of the hypoglycemia. So you can see that when you have the simple glargine here, when you inject a big dose here, the insulin will go to very low, you know, somewhere around 40 milligram per deciliter range. But the smart glargine here, you will just kind of stay a little bit higher than a little bit higher than 100 milligram per deciliter, even and then without the induction of hypoglycemia. So basically, this is to show you that this insulin can indeed do this the way we would like it to. And then so we are trying to further improve this one, the way we think there's some room for us to improve is that right now, you can see that even in the hyperglycemic range, it's only the same activity. So basically, the setup point is somewhere around 300 to 400 milligram per deciliter. We are trying to basically make it in the way that somewhere around 125 to 150 milligram per deciliter, the activity between the control one and the smart one is the same. So that will be the case that we can basically potentially, if we want to make the optimal insulin, we want to see around 400 milligram per deciliter. This gray bar here will be way higher, and then still the same. In 100, we'll say 70 milligram per deciliter. The gray bar here will be lower like this. And then we recently just got a R1 for us to work on this, and then hopefully in a few years we can work on achieving this goal. And then another approach is we are trying to do it, and then because of time, I don't have enough time to show you that. But basically, what we are trying to do is basically we can also try to constrain the insulin conformation such that its binding toward the insulin receptor will be glucose dependent. And then so basically that we can basically modify insulin such that we can create some of these specific artificial constraint to do that. And then very recently, just earlier this week, we received a pilot grant from the Diabetes Center here at Stanford. I'm trying to work on this, and then hopefully we can have some progress on that in a few years. And now I'm going to switch gear to talk to you about some of the developments we have about fast-acting insulin. So I mentioned to you that in the early days, people need to do multiple injections per day, but because of the advance of the continuous glucose monitor and the insulin pump a decade ago, and then also a few years ago with the hybrid closed loop insulin delivery system. Now things are much better for people with type 1 diabetes to basically control their blood sugar levels. So now basically there are algorithms that can do a very good job to help people with type 1 diabetes in a way that other than the bolus you still need to do for the meal time, but throughout the day, they are pretty good controls there. But to achieve even better, the so-called artificial pancreas, there could still be some areas we can work on to make things even better. There are some engineering problems to solve, but some of things that still something we can pursue think about is, in the normal healthy, in the normal pancreas, in addition to beta cells to secrete insulin, there are still alpha cells to secrete glucagon. And then part of the reason why the glucagon pump is very hard to do is because of the glucagon stability problem. So there are people trying to work on that in a way that we can have a bi-hormone system for the artificial pancreas system by achieving a more stable glucagon formulation. Another potential way to improve it is an even faster insulin action. This is because when we do injections of subcutaneous insulin, it actually takes time for insulin to reach the bloodstream to have the effect. And I will talk deeper about this later. Here's a slide to show you that. So basically, because we are doing subcutaneous injection instead of IV injection, so insulin is not really achieving the bloodstream right away. And then insulin normally forms a hexamer, and there are good reasons for that. That's because insulin is a tiny, and our beta cells make a lot of it. And then normally tiny proteins are not very stable. And then for insulin to be stably stored, it basically evolutionary, becomes a hexamer with 2 zinc ions for the coordination. So this is basically a good thing for insulin to have this. in normal situation, hexameric insulin will be released to the bloodstream, and then basically things will be diluted out. Normally, it's somewhere around 2 digits picomolar insulin concentration, and then that's way below the dissociation constant for native insulin. So in the bloodstream, there are always just monomeric insulin. But when we do subcutaneous injection, it actually takes time because normally insulin in the vial is somewhere in the millimolar range. When you do subcutaneous injection, because of the subcutaneous fat we have, the dilution factor is at most, somewhere around 10-fold. So that means that it still takes another 10-fold dilution for insulin to be dissociated into monomeric form. And then even when they are, the so-called fast-acting insulins, the dissociation constant is somewhere still around the concentration we will have in the subcutaneous depot. So in those cases, that means you will have a mixture of dimer and in the monomer. So things will be faster, but not fast enough. So one can imagine that if you can have something that will be, monomeric to begin with, and then things can potentially go to the bloodstream faster. So this is something we try to achieve in a way that can we make monomeric insulin to achieve ultra fast acting properties. And then I mentioned to you in one of the earlier slides, talk about the insulin development, that there are a lot of ongoing efforts to achieve even faster acting insulin formulations, mainly still using the same insulin. So the homolog or Novolog, but using different excipients. And there are quite some progress there in a way that, so Novo has a product already in the market, and then Eli Lilly recently get some good news from their clinical results as well. They are using a vasodilator, so as an excipient, so such that the vessel will swell bigger, so such that the hole will be bigger for the insulin to travel in. And then Professor Eric Apple in the material science engineering department here at Stanford, they are also developing a lot of great polymeric excipient system to achieve the same goal. And then the way I imagine here is basically, if we can have a monomeric insulin, we can potentially get something even better if we can combine with some of those advances in the excipients area to achieve something even better. Here is actually some of the interesting here. So basically, to achieve monomeric insulin, the bigger problem is we need to break out the insulin dimer. So what I'm showing you here is the crystal structure of the insulin dimer interface. So as you can see, it's a pretty strong insulin dimer here, okay? And then this is a insulin called insulin aspart or Novolog. This is one of the early fast-acting insulins, Novo Nordisk, they are developed. So what it does is do one mutation to change originally a protein residue in insulin here to be asparic acid. And as you can see here, because now this is a negative charge group, now this is another negative charge group. So they create two amino acid repulsive pairs, so such that the dimer will be weaker here. But you can see that this is the bulk of the dimer interface, and they are actually not changing that. And there's actually, because there's a good reason for that, because this is the part involved in the dimer formation, but this is also the part involved in the insulin binding. So if you want to mess up with the area here, you will lose the insulin bioactivity. So such that they can only play around in the residues here. So they can make something, between monomer and dimer, but it's not really monomeric. So this is just what I talk about, because these FFY, so the residues here, they are so important for the insulin binding. You can cut them off and make an insulin like this. We call it DOI because you cut the amino acid off. So the whole thing will be monomeric. But the problem is 40 years ago, so 4 decades ago, we already know that when you cut them off, you will lose the potency by a thousandfold. So basically, this has been kind of the dilemma in the field I mean, basically, we know the structure of insulin. We know so much about what insulin is doing over the years. And actually, there are somewhere around 4 to 5 Nobel Prizes related to insulin. So this is one of the most well-known and well-studied proteins, but basically it's a dilemma for people to have a monomeric insulin because it's just not possible for you to cut the dimer region off to maintain the monomeric form, but still maintain the activity. And we, like others, don't have a good way for that. But as David mentioned, we are lucky to get an inspiration from the corn snails. So as you can see from this video, so basically, these are corn snails, and for this specific one, it's a species called Conus geographus. And then as you can see here, the snail just opened the mouth and then to eat a fish. And then normally it doesn't make sense because fish should swim fast and then just escape. But as you can see here, the snail just it's doing something in a way that the fish won't move, and then such that you can finish the prey hunting purpose. So there are a lot of people trying to study why these snails, or the fish hunting corn snails, can eat fish. And then so in the old days, people were kind of chop the snails off and then try to extract what's out there and then try to know what's going on. A lot of times they will get a lot of ion channel blockers or neurotoxins, and that makes sense because what the snail has to do is to intoxicate the fish and such that the fish will stay there for it to eat. But this time, from our collaborators in the biology department at Utah, they found a molecule like this. And then if you are someone like me who worked with insulin for a long time, you will see that this molecule from the snail, it looks like human insulin. So this is a venom insulin from the venom of the snail. And then just to tell you, this is not the endogenous insulin snail use, and potentially this is a venom insulin it developed to target the fish receptor to make fish not to move. So what is special about this venom insulin is exactly that what I previously talked to you about. It has a very short insulin sequence, and it doesn't have the dimer formation region. And this is basically the reason why we decided to work on that. And then what's special about our lab is basically we can use chemical ways to make the insulin at will, and then we can use this strategy to make all different kinds of insulin for testing. And this is just one of the original studies they did in a way to prove the mechanism. So one can make zebra fish to be diabetic by treating the same drug I talked about to kill the pancreas. So when you you can have diabetic fish. When you add fish insulin, or you add this venom insulin, so you can see that the blood sugar level will drop down. So the venom insulin indeed work in the fish receptor. And also from this video, so you can see that in the control fish, you will swim very fast, but in the fish with the venom insulin there, you just won't swim. So it's a way to prove the mechanism. So I mentioned to you that because this venom insulin doesn't have their dimer formation region, so one we expect it to be monomeric, and this is exactly what we do. So we did it, and then we use actual neurocentrifugation to prove it. So the association state is 1.05, so that means that only 5% of the species is in dimer, and then the majority of the venom insulin is the monomeric form. So that means that the hypothesis was right. And but it's good in fish receptor, but what we really care about is its activity in the human receptor. So we did both receptor binding in human receptor and then see the receptor signaling. What we are seeing is the snail venom insulin is somewhere around 24 weaker in terms of the potency compared to human insulin. So it's for sure not great enough for further clinical development, but it's pretty encouraging because I mentioned to you that normally in the human insulin case, when you cut the dimer formation region off, you lose the potency by 1,000-fold. So the fact that it's only 23 in potency, that means there are more for us to study. And that's why we get to study that by using structure biology techniques there. So the main idea I want to show you from this beautiful figure is that normally human insulin, it needs a dimer formation region, or more specifically, these originally B24 phenyl alanine residue here from the black color here is the key. And for sure, the venom insulin, because it got cut short here, so it doesn't have this. So for the venom insulin to have the effects to compensate for this role, we believe from the structure there are two mutations here. One is from B15, one is from B20. Originally, they are leucine and glycine here, but in the venom insulin, both got mutated to tyrosine, and potentially one or both of the tyrosines will compensate for the effect here. And then this is from 2016, and then we have been studying that, and then we very recently we just published this. So I'm just going to tell you the result here. So in the end, what we have is we generate insulin. We call it mini-insulin. So you can cut the dimer formation region off. So you can see the B chain is short. So the whole thing is monomeric. And we are putting in 4 mutations in red here: H, R, E, and Y. And as you can see, all 4 are basically what we learned from the Venom insulin sequence. And now in this mini-insulin with the 4 mutation, we can basically move the potency all the way from 1,000-fold weaker in potency in all the way to basically very close to human insulin activity. And then this is a busy slide, but just to tell you that the mini-insulin we have, signaling-wise, binding-wise, it's behaved very similar to human native insulin. And the 4 mutations we put in, they are all important because we remove one of the 4, we will basically reduce the binding affinity somewhere between 4 to 20-fold. And then because this is published, so I'm just going to tell you what's happening here. While we end up figuring out mechanism-wise, what we believe is there are 4 mutations. They are doing different things. So one of it is what I just told you from our result 4 years ago. The B20 tyrosine is compensated for the role of the missing dimer formation region here. And then in the 3 other mutations, we are introducing 3 new saw bridges to further increase the affinity. So basically, this region is this black region is very important, and you need to have that. But throughout the evolution, just the wonder or the magic played by the venom insulin, they can have 4 other things. They just evolve in a way that we can learn from that to go, to overcome the dilemma, to achieve this kind of monomeric insulin with the same native bioactivity here. And then what we are trying to do, and then I will say part of the big reason why moving to an environment like Stanford is really amazing is we would like to basically test all these molecules in pig studies here. So early on, we kind of collaborate with the Apple lab here at Stanford in some of the preliminary study to basically test how active or how fast these mini insulin can be. So this is just very preliminary result here because we will need to do much more here. But as you can see here, our mini insulin in diabetic pig, at least in the first 30 minutes, we can start to see some significant increase in the blood glucose dropping actions here, and then this is quite exciting. Potentially, we would like to test more in the near future here. But there is actually one problem about monomeric insulin. This is that I mentioned to you that there's a reason why native insulin would like to be hexameric. That's because normally monomeric insulin is not stable, and that's the thing here. So when you just put native insulin, okay, so you will still have a tendency to form dimer and hexamer. But if you don't add zinc, don't have it here to coordinate. And then when you put it in the vial, shake it, and then 37 degree, somewhere around 10 hours, you will aggregate. So when you see it hit there, so that means the whole thing will form kind of fibro type of stuff. And then that's not good. And in the mini insulin case, it's actually even worse. You will form aggregation in somewhere around 2 hours. So this is for sure a problem for us to address. And I will talk to you later about how we are going to address this. Last year, we actually published a paper to basically about the discovery and the characterization of even more venom in insulin here. So one of the comments we got from people is, the story of what I just presented, they are great, but that's just one example. And it could be that those are designed for something that's not really insulin, and then that just happened to be something that works for fish receptor, but that may not be the purpose. But now, as we showed last year, we can now find 6 more new sequences from 3 different species. They all have insulin-like skeleton and potentially using different mechanisms here, and then to achieve various binding affinity toward the insulin receptor. And then so this is basically to tell you that this is most likely a real mechanism these snails are using to kill fish, because all different species, they all share the same thing here. And then one of the things we discovered here is one of the venom insulin here, it has a longer A chain. And then from our model, it seems like the region between this part of A chain and then somewhere around the end part, the B21, B22 part of the B chain, they are quite close. And that's why we decided to study it. And then basically we see that they are actually quite close, and potentially we can form a 4th disulfide bond. So normally there are 3 disulfide bonds in insulin, and this is something that for us to have a 4th one. So basically we can see that we can actually form a 4th one here, and then without sacrificing the activity. And then we solve the crystal structure here, and then basically the 4th one basically just beautifully designed here without really changing the backbone of the insulin. So what can these 4 disulfide bonds do? It can actually make the whole thing much more stable. So when you put a 4 disulfide bond in native insulin, you can make the normally 10-hour aggregation time to somewhere around 40 hours. And when you put it on the mini insulin, normally it's not stable, but because of the 4 disulfide bond we are putting, that will probably make the whole conformation to be even more stable. So so we can see that this mini insulin won't even aggregate as long as 100 hours. And then this 4 disulfide bond also give us the opportunity to further study whether there are potential new mechanisms we can use to discover even more mini insulin with monomer properties here, because this is pretty different from the previous ones I talked to you about mini insulin, that in a way that it doesn't share the same mutation. So this guy is using a totally different mechanisms. And then I'm just going to same way kind of show you the results. So so this is pretty new here, and then we are about to submit this result. So basically now, instead of having mutations here like what I showed you, this new mini K1 insulin, basically all you need to have is 3 more amino acids in the end of the A chain, and potentially these residues will compensate for the role here. And then this leucine is very critical, and I will show you there to tell you what happened. And then we also do some structural active relationship to kind of know what's going on here. And then what's even more exciting is this mini insulin itself is already pretty stable in a way that basically it won't aggregate after 100 hours. So this is going to be a pretty exciting candidate for us to study. And really what's going on here, we actually solved the structure of this insulin with the receptor complex. And then, so basically we have a 3.6 Angstrom resolution using Cryo-EM. So this is the activated receptor, and then the dark gray color here is the insulin here. So in terms of binding and activation, it's very similar to native insulin activation. But really what is special here, I told you that we all know that native insulin is using this B24 phenylalanine here for its effect to really bind with this part of the receptor. And this mini K1 insulin it's using this leucine, and as you can see here, this leucine is occupying exactly the same binding pocket here. And then basically this proves the mechanism for us that basically this is another pretty amazing magic the venom is, or the snail is playing in a way that this is something we can never imagine or design if we try to put try to design it, but really learning from the nature, we can actually we can actually discover something like this. And this, I would say, this is just the beginning. We have the, we have the Hung-Chieh case and now the K1 case. And then I don't have the sequence to show you here, but potentially there are way more things for us to study, and they are all using very different mechanisms. And then basically, they are just more nature for us to explore here. And then this is another way I will say, basically another reason for us to believe that there's really a great reason to study more basic science, because for these people originally trying to study why the fish hunting cone snails will kill fish, they don't have too much in their mind, but eventually by studying more and more, and there could potentially be some more potential very useful things we can learn, and then potentially help to develop better clinical products for people with all different kinds of diseases. And then finally, I would like to acknowledge the people who are doing that. So the whole project is spearheaded and championed by my postdoc, Chou, who moved with me here in Stanford. And then Maria Desotra, she was my first graduate student, and then she is now a scientific director at DOD, and she provides a lot of great insights here. And then Gabby Gabash, who is a technician in the lab, and she's now a medical student at Utah, and then she provides a lot of help as well. Ellen Blakely is a joint graduate student, and she stayed in Utah. And then these are my current lab members. And then Professor Savadi and Professor Oliveira, they are the key marine biologists, and they are the main people who discovered the original sequences. And then my lab worked a lot with all the various type of people. So we work a lot on structural biology to try to know the structure, to understand why. We also work with a lot of people to work on the to prove that these insulins are active. And then these are the people that help us to do various biophysical and the biochemical studies. And these are our funding support. And thank you very much for your attention today. I'll be very happy to take any questions.
Thank you very much, Danny. That was a wonderful talk. It's really fabulous to see how you are using basic science to translate these discoveries into therapies that are going to benefit our patients. The cone snail story reminds me of the Gila monster saliva story, which was about 15 years old, I believe now, when it was discovered that the Gila monster saliva had homology with GLP-1. And over the course then of probably 10 years, the development of the GLP-1 agonists, which are increasingly important part of therapy for type 2 diabetes. So the cone snail story, we are excited about that, and really looking forward to next steps with your work. So we'll take any questions that come in, and we've got a first one from our colleague Daryl Wilson, who asks, is there any information on antibody formation in response to repeated use of the mini-insulin analog?
Great question. So in our paper this year, so only on the mini-insulin one, so not yet for the mini-K1 and some other stuff. So we do an animal study that is a 21-day straight injections to B6 mice, and then we don't see antibody formation, and we don't see T cell proliferation. This is about what we have done so far in terms of looking for immunogenicity, but the caveat here is these are models in a way that you will only see positive. You will only see you only see the result if it's severely immunogenic. So we don't see that. So that only means it's not severely immunogenic. And then I would say one will argue that this will be something that you will need to do some human studies to know, because the mice immunology is probably quite different from the human one as well. The way we have been thinking about it, and then we have some industry partners for us to discuss this about. So basically, what we are thinking is, if the mutations is not in the range where they are basically known segments that we know that it's involved in the kind of type 1 diabetes, the immune response range that will normally be fine. And then in the case of our thing, the mutations we are putting in, it seems that they are outside. So that looks to be okay, but I would say that all these things you will need to do more human studies to know.
Great, thank you. And just looking, we just had another question come in. So many cytokine receptors use different ligand isoforms to the same receptor to yield subtly altered signaling. Besides glucose control, is there any difference between downstream insulin receptor signaling between native and engineered insulin?
This is a great question. And then, so from one of the slides, I show you that basically these, at least for mini-insulin, it doesn't really differentiate the two signaling. So I mean, so say for the case of insulin signaling, it could be more toward the AKT phosphorylation way or toward the MAC-ERK way. So basically, these are the more metabolic arm or the more mitogenic arm for insulin. So in mini-insulin case, you don't really change that. But this is a great question, because we actually have a NIH grant trying to do this as well. So basically, one would actually like to generate insulin that will more to a metabolic arm instead of mitogenic arm. And then we believe there are ways to do that, but we actually don't have a lot of progress there yet. But I will say this is something people really want to do. And then so the cytokines, and then also there are some other things related to this as well that provides some very exciting examples for us to learn. And then I know that people very rarely talk about this here, but I actually recently talked about this in our divisional kind of meeting. For insulin receptor, there are actually different isoforms as well. They are just not very well known. And then for native insulin, it doesn't really differentiate the two. So one way to address what you just talked about is potentially to develop isoform selective or isoform specific engineered insulin to test that. And this is something that is very hard to do as well. But as I just show you that some of the things we learned from the snail can overcome a lot of these traditionally very hard to do topics. And then what we believe is mini-insulin can actually start as a model skeleton for us to develop something like this. Yeah, so this is a great question.
Great. Well, thank you, Danny. It looks like we are just at 9 o'clock, and perhaps good to wrap things up so people can get on to their next Zoom or possibly see patients or whatever else is in the day. So Danny, just want to thank you again for a wonderful talk. Thank all the audience for their participation and being here with us today, and really looking forward to ongoing work with you in the years ahead. So thank you very much. And thanks everyone. Have a good weekend. Bye-bye.
The preceding program is copyrighted by the Board of Trustees of the Leland Stanford Junior University. Please visit us at med.stanford.edu.